So good morning, everybody. So I would like first of all to thank Giovanni for inviting me, and also I gave him a couple of options for the presentation subject, and he picked up this one. So if you don't like it, you can complain with them. So anyway, okay. So the focus is about SMYD3, which is Lysine Methyltransferase. We have been working for a few years on this target. Even if I'm the presenter of this work, actually the work is the result of a multidisciplinary team, which involves not only my group of University of Bologna. So here you have a list of people. Some of them are not anymore there, but they moved to other institutes, such as Institutet, who is now not. It was a former postdoctoral fellow at my lab, and now it is working as a researcher at the CNR. And also it is a strict collaboration with the group of Professor Elena Danielsson from the University of Uppsala, Sweden. She will show you the part she took care on, and actually without her work on this subject, we would never be able to do this work. And then again, it is a contribution of people from ESOF, CNR, as well as from company which is called Innovamol, led by Dr. De Rio. And for the biological part, cell-based assay, we involved people from the Bellis Herx Institute in Bari. So it is a great network, and actually here it is just summarizing those people who were involved in this specific project, but actually the network also involved people from the University of Milan and Rome, because actually there is sort of SMYD3 network. So I'm just the presenter, but obviously I did not do the job by myself. So here it is, just to briefly give you why we were interested in SMYD3. So SMYD3 is a potential epigenetic target, especially it seems to be important for colorectal cancer, but now it seems also to be involved in breast cancer, either female and male breast cancer. We have been more focused on SMYD3 in colorectal cancer because there was background interest from the people from the Bellis Herx Institute in Bari, which have been working on studying this enzyme as a target before I was involved in the project. I must mention why it was important, and my group was involved in the project. So I'm leading a laboratory which is called Pharmaceutical and Biopharmaceutical Analysis Section. So our main expertise is to develop tailored assays in order to characterize new compounds. So we have several expertise in some biophysical techniques, as well as in chromatographic techniques, spectroscopic techniques, and mass spectrometry. And so depending on the target, if there is no commercial assay, or if the commercial assay is not satisfactory, it might lead to some false negative or false positive. So actually there are doubts in its use in early phase of drug discovery, so correct prioritization of compounds. Then it might be useful to have a tailored assay in order to proper screen and try to avoid as much as possible such a kind of its selection, which then will end in not proper it compound. So here is the enzyme. The enzyme is a methyltransferase. So actually it works thanks to the presence of a cofactor, which is SAM, S-adenosine methionine, with the delivery methyl group, and as a substrate is specifically methylating some lysine residues in histones, which are the primary targets, mainly histone 3, but also histone 4. Why it there is an interest on this target is because actually it is known that it is overexpressed in cancer cell line, and is depletion, actually depletion of expression does not have functional impact. So actually it might be suppressed by a strong inhibitor, and supposedly there should be not highly toxic effects. So it is treatable, but actually when we stepped into this work, this was and is still under validation as a target. So I must mention that most of the works that have been published on this enzyme are mainly related to probe development more than drug development, because actually those probes were needed to prove the involvement of this enzyme in cancerogenesis. Now the understanding is going to be wider, and actually it seems to me say that there is an just accepted paper in which it is shown that actually likely this enzyme is involved in is overexpressed in cancer cells because it grants that the DNA repair system works properly. So it is overexpression of these enzymes a way of ensuring the cancer cell to be protected by DNA damaging agents. So by inhibiting this enzyme, actually, it is likely that actually it is proven in that article that it is shut down the repairing system. So actually the cell became more sensitive to the DNA damaging agents. So then actually the death is due to the DNA damaging agent, but so you can plan for a conjoint or co-administration therapy. So this is just to locate the target. And so here the focus. The setting, so the focus is to show you how the platform for screening for SMYD3 inhibitors, especially this platform that led to the discovery of a new allosteric binding site and allosteric binder, was set up. I must say that actually being involved in different screening campaigns due to the involvement of several groups, actually several different assays were developed in order, and they were used conjointly in order to understand the mechanism of action of those new compounds, which were selected either based on computational studies performed mainly by Novamol, as well as based on historical chemical libraries available at CNR, at the CNR group. So here just briefly, what does it involve? So I may say that to have a good screening tool, you need to have group, so actually, let's make it even further. To have good results, you need to have a good screening tool or more than one screening tool, but also you need to have a good enzyme to be used for the screening. And this was not the case for SMYD3. So after a while, we were screening different commercial sources. We end up in concluding that none of them was suitable for performing reliable studies. Either the activity was very low compared to the amount of enzyme provided. So actually most of the enzyme was not active. So it means that you may end up in having your inhibitor specifically bound to whatever is not active, so you don't see functional activity. Furthermore, most of the enzymes available on the market, and that is still are produced with GST tag. Now you have more ACE tag available, but GST leads to many specific interactions, especially if you work in SPR, so you have a huge signal. And so everything in combination made us stepping back and going to set up a cell expression system in order to get active and properly working full-length SMYD3 to use for the assays I'm going to present. So this is an important step, and it was made by the group of Professor Elena Hjalmarsson in Uppsala. That's why I said without her help, nothing could be done, could have been done. Then another important step, we have two. I'm just briefly summarizing here because I'm not going to speak all of those due to time restriction, but most focusing on the bioanalytical platform, actually biophysical platform. So we had also to develop a proper new assay. The assays that are available for the functional activity, for the evaluation of the functional activity of SMYD3 are either based on radiolabeling or radio compounds. So actually you need to work with radioactive compounds, which is we are not allowed to, or tandem assays, which means that you have two enzymes, and then you always wonder if your activity is due to the primary enzyme on the report enzyme. So since we have an expertise in mass spectrometry, actually we developed a specific MS-based assay. So using EasyQTOF mass spectrometry system, we could monitor directly using limited sequence, which is the targeting methylation sequence of the histone 3 sequence. Actually, we could monitor directly the methylation, and all the validation was published previously to this work, so the assay was fully validated. Then, what is actually the focus of this work is, in this talk, is on the platform, mainly based on SPR, surface plasmon resonance. We developed in order to try to screen compounds based on their affinity, but also to understand where they were acting. And then we end up in that allosteric compound. So here is just what I just explained. So actually the starting point was to step back, having another expression system, which was able to provide a properly functional, suitably functional, and good amount of SMYD3, full-length SMYD3 enzyme, and also an activity assay, which was not based on radioactivity, neither on tandem enzymes use. This is just to show you that the stability was proved and confirmed by using fluorescence thermal shift. But actually, this is a very easy way of proving that what you have produced or isolated is still keeping the conformation and the proper conformation and ability to bind ligands. So actually you can monitor the melting point. It is very quick, you don't need to label. So actually it is very quick system. And so by confirming the stability of the enzyme after binding substrate, cofactor, etc., you can confirm that actually your enzyme is functional. And usually it is often combined with SPR assay, because actually in this way you can prove that what you are going to mobilize in the sensing surface on the sensor chip is active before immobilization. So you are not going to immobilize something which has lost conformation and activity. Those studies were obviously combined with activity studies in vitro using the MS-based assay that I just shown you. Then for screening, we select the SPR technique, surface plasmon resonance technique. I guess most of you might already know what is this kind of technique based on. However, just to briefly summarize, it is microfluidic based systems in which optical system in which the binding and this, the association dissociation phases of biorecognition phenomenon can be monitored in real time. The system is based on the use of a sensor chip, which might be divided into 2 or 4 or more flow cells, depending on the type of instrument you are using. Usually the minimum number is 2, a reference cell and a sample cell or a working cell. the enzyme of interest is immobilized on the surface. In this case, the enzyme SMYD3 was immobilized using a tailored immobilization system, immobilization protocol using classical amine coupling. Modification was required because this enzyme is not so stable, so either the mobilization condition and the surface deactivation condition were made milder, so without any strong use of strong reactant because it would damage the enzyme. And mobilization was done at a lower temperature than room temperature, so at 15 degrees, which ensured the enzyme to be stably immobilized and was fully functional. Activity, function, the fact that the functional enzyme was immobilized, so the enzyme immobilized was functional, was proved by monitoring or assessing the binding profile for SA, which is a substrate, and SA, which is the product. So while the substrate is bound, is typical profile in which several concentration of substrate, in that case cofactors, sorry, are added, and actually the dissociation is quite low, while the product obviously has a fast association and fast dissociation kinetics. Those data were in compliance with what was reported in the literature, and then other known inhibitors were tested, but I'm just showing this one because this one was the inhibitor we screened and that then we decided to use for further step. This is known commercially available inhibitor, reversible inhibitor of SMYD3, developed by the company Epigenetic, which has led many screening campaigns on this enzyme. What is peculiar of this inhibitor is that, apart from being quite potent at nanomolar level, actually, just to try to, these are, this is the association and multiple injection in the assay. And here is the dissociation phase. And as you may see, it's easy to understand that actually dissociation is almost negligible in the time frame of, in this case, 1,000, yeah, more or less 1,000 seconds. So it means that this inhibitor binds, is reversible, but it binds to the substrate active site. And it's the kinetics of dissociation is so slow that actually it takes many hours to significantly dissociate. So it is reversible, but it is really long-standing inhibition. So in terms for in the time frame, we usually perform assay studies, assay analysis cycles in SPR. Actually, for the time frame of a typical SPR cycle, this inhibitor actually it gives us irreversible inhibitors. I'm saying that because this finding was very important for setting up the screening system. So we the screening was SPR. So the instrument was an SPR system which allows to work with four flow cells. The experiments were made by Dr. Fabini, who was my previous postdoctoral fellow. Now is working in Evotec UK. And actually, what he did, he decided to prepare these four flow cells in this way. So two were reference cells. The reference cells are this one and this one. So one is just the surface without any modification, and this flow cells means to account for non-specific interactions with the surface, the sensor chip surface. While the second cell, the second flow cell, contains was in that flow cell, we immobilize the denaturated SMYD3 enzyme. And the aim of this cell is to account for non-specific interactions with the enzyme. So to rid off of all those small molecules which were able to interact in a specific way with the enzyme surface just because of hydrophobic or labile interactions without any specific binding to an active site or allosteric site. Then two active cells, which are the one in which SMYD3 was immobilized, and then another cell in which SMYD3 was immobilized, and then just on that cell, we injected the epizyme inhibitor I've just shown you in the previous slide. So actually, in this cell, we have the inhibitor binds to the substrate active site and keep the active this site occupied. So actually, here the SMYD3 is not anymore available the active site for binding any other ligand. So what we can expect as outputs is that we have compounds binding here and not here. It means they are compounds that are binding likely to the active site, substrate binding site. And since in the following flow cell this binding is occupied, then they do not bind anymore. Okay, or we can have compounds that are binding to both active flow cells. So it means they are binding to some other binding site. And so the most likely one is the cofactor binding site. To prove if it was true, the same set of experiments were performed by adding or not adding in the running buffer some, so the cofactor. So then we have another two options. Then the activity, the binding is kept when we have some in the running buffer or is not anymore kept. So if it's kept, it means that the ligand is binding somewhere else. If it's not kept, it means that binding is at the cofactor binding site. In this way, we can distinguish in this simple setup where is the binding site. So if the new compound or the binder binds to the substrate binding site, to the cofactor binding site, or somewhere else. So just since I have just 5 minutes, here is the results. So here we use SA as a control, just to make sure that it is a quality control, to make sure that everything was working properly. 45 small molecules were selected, and actually, obviously, we screen also in other system, but here what it is important to see is that is standing out. It is the identified compound, which is a commercial one called diperodone. And actually, this was able to bind to both flow cells in the presence as well of the cofactor. So it means it is able to bind SMYD either even when the binding substrate binding site is occupied, and even in the presence of the cofactor. So it means it is able to bind specifically with no signals in the reference cells in the presence of when both cofactor and substrate sites are occupied. So it should bind somewhere else. So to make the story short, its affinity constant was evaluated by SPR, and actually it is not a strong inhibitor. Its KD is in the range of 60 micromolar. The one compound that was inserted in this library, the small library, is a racemic mixture. So also then comes the question if one of the two enantiomers was more prone to bind compared to the other, or there was no enantioselectivity. So Dr. Tedesco set up the enantioselective separation, preparative separation of the two enantiomers, and identified the two peaks in terms of S and R by combining theoretical studies with CD experimental acquisition of the two spectra. And so we identified which peak was related to S and which one was related to the R diperodon. They were both tested again on the same screening system to assess for their eventually enantioselective binding. However, difference in terms of affinity was very low, 42 and 84. So actually there is a slight difference, but not really significant in terms of biological activity. In parallel, no significant difference was encountered in their activity. So they bind similarly, they are active at the same way. Again, in line with their low affinity, also the activity was quite low. And then crystal studies were performed by Dr. Vladimir Talibov. And at the end, the studies show where the binding site is located. So just for the following slide, just remember that the white part here in the picture is the C-terminal part of the enzyme. And this part seems to be involved in interaction in binding with HSP90. And this binding seems to be involved in the translocation of this enzyme, as well as in triggering some functions. So, and the location of the binding site is just here at the edge of the C terminal area. So when we found this location, here you have here are the two enantiomers bound. They both can bind in the binding site, in this allosteric binding site. Here are the two different modes of binding. The binding site, the allosteric binding site, which was identified, is pretty permissive. So obviously, this is not a ligand which was developed based on the binding site. So there is space for improvement. Just getting to the final slides. So since the position of the binding, the allosteric binding site is partially overlapped to the region which seems to be involved in interaction with HSP90, then obviously it comes to our mind to prove if there was some kind of inhibition of that interaction. So we set up another SPR-based assay in which either SMYD was immobilized and HSP90, a part of HSP90 was screened, or HSP90 was immobilized and SMYD was screened. So used as an analyte. So this proved that they actually then can interact independently from which target is immobilized, and their affinity is around 13 micromolar. However, when we screened the diperodon, either racemic, either enantiomers, we could not see any significant inhibition of the interaction. This might be obviously due to the low affinity and also to the partial overlapping, but so it was good to try if any inhibition would be seen. And just this is, I guess, this last slide of our conclusion, just to show you that then the either enantiomers as well as the racemic mixture was tested in cells. I'm just showing this was made by Irsi in Bari, and actually it is they were trying to see if a chromatin association of SMYD3 was impacted by the presence of diperodon. This is related, so the translocation of SMYD3 and binding to chromatin is related to its association with HSP90. So actually, if any effect would be seen, it means that actually this interaction will be affected. And this is an interesting, but we have no answer, so I'm just showing the results. So actually, when we use the racemic mixture, it's the binding or accumulation of SMYD3 on chromatin was much reduced, while when we use the two different enantiomers, there was no effect. This may be due to the fact that both enantiomers also can bind to the same active binding site and active site, binding site. It might be that, binding simultaneously, they are covering a larger surface, so they have a higher impact on this destruction of PPI interactions. It might be an hypothesis, which obviously it is just an hypothesis, should be proved, but it's the only hypothesis that came to our mind. But obviously, we are open to any other suggestion. So here are the conclusion. Hope I've been in on time, maybe. I don't know. So late. Okay, so speeding up in conclusions. So here, so just to say that I try to show you more than impact results, the possible workflow that can be used also for other enzymes to try to understand if you're looking to mode of action or try to select new compounds or new potential inhibitors acting on a specific site. I hope to have shown you that this might be one of the options you can go through in order to modulate your tailor your assay in order to have one site available and then to understand where your system binds before going obviously to dedicated X-ray studies. And the system was enough sensitive to also highlight binding of compound, which was quite weak. So there is not problems in sensitivity. And so this new binding site, obviously this inhibitor, this binder is not optimized for the role, but it might open the a new line for being optimized and try to have an optimal binder for this new allosteric site. And with this one, I thank everybody for your kind attention. Sorry.
Thank you, Manuela, for the very interesting talk. For questions.
Maybe I have one question. I probably mentioned it, but I might have missed it. What is the throughput of your assay? So how long does it need to test one?
Single screening in duplicated 7 minutes.
7 minutes per comp.
Yeah, it depends mainly, if you have a binding and the activation phase. So if you screen a single concentration, it's very fast because you are speaking about tens of seconds and tens of seconds. Then you need to regenerate your surface. So it is few minutes. If your binders are a little bit more resistant, you need to do double cycle regeneration, so it takes more. But we are in finished for each compound.
Can you parallelize the sun?
No, sorry, I was saying that if you need to make a KD determination, obviously it takes more time. Usually, I would say a couple of hours, depending on how many points you need to screen.
And can you add more sensors in parallel?
Sensor you need many.
Parallelize it, yeah.
Yeah, depends on the instrument. So if you have an advanced one, you can have even 8 or 16. So you want it to have so many.
Right. So please join me in thanking Manuela again and all the speakers from the